Hello, everybody. Welcome back. This is the first lecture in our Chapter 5 content. This is a mini lesson that's going to cover the epidermis. But before we get too far into that, I wanted to talk about the integumentary system as a whole, since this is our first lecture in the Chapter 5 material. So the integumentary system includes the cutaneous membrane and accessory structures. So that's kind of our two big components, but we can dive into that a little bit further. So if you remember kind of the middle of chapter 4, when we got done talking about connective tissues, we mentioned 4 types of membranes. So we had mucose membranes, serous membranes, cutaneous membrane, and synovial membrane. So the cutaneous membrane, it's a membrane because it has an epithelial component and a connective tissue component. And so that is your skin, basically, is your epidermis and your dermis. That's the connective tissue component is the dermis. The epithelial component is your epidermis, so we also can call that the cutaneous membrane. The other things, and here's kind of a picture of our integumentary system. So we can see the epidermis here. Right? That's the epithelial component of this membrane. And then here's the dermis. That's the connective tissue component of the membrane. And then we also typically talk about the subcutaneous or hypodermis with the integumentary system, but technically it is not a true part. It's just one of those layers of fascia, right? So we went through all the different layers of fascia in the connective tissue part of chapter 4, but it is technically not part of the integument. It is the integumentary system is the cutaneous membrane, which is the epidermis and the dermis, and our accessory structures, which is hair and glands. So sweat glands, oil glands, and what's not on here are nails, like fingernails and toenails. So those are our accessory structures. So I'm going to present the content of chapter 5 broken up into these little mini lessons. So this one's going to cover the epidermis. I'm going to have one talking about the dermis, and I'll have a mini lesson for each of the accessory structures. And then I'll have one at the end talking about damage like burns, skin cancer, and repair. How does your skin repair itself? Because we know it does. It's one of those wonderful qualities of both epithelial tissue and connective tissue, and they have those regenerative abilities. So let's focus first for the rest of this lesson on the epidermis, the uppermost layer of the cutaneous membrane, however you want to call it, integumentary system, outermost layers. Okay. So your epidermis, the type of tissue that it is made of, falls under the epithelial tissue category. And then hopefully you're getting really comfortable with your tissues. We can identify the basement membrane and the free edge. And we know there's multiple layers of tissue, so it's going to be a stratified type of tissue. And if we look at the cells at the topmost layer, they're going to be flattened cells. So your epidermis is composed of stratified squamous epithelium, okay? And in this particular, this skin, like your dry skin, this is what we call keratinized stratified squamous epithelium. Compare that to, say, the lining of your mouth, right? Like your cheeks and stuff. Inside your cheeks, that is non-keratinized stratified squamous epithelium. Keratin is a kind of thick waterproofing protein that helps to hold the cells together and makes them waterproof. It allows our skin to be a dry membrane as opposed to the wet membranes of like mucose and serous. But it's still made up of stratified squamous epithelium. Now, if we take a closer look at this stratified squamous epithelium, we'll see that there are multiple layers within that one type of tissue. So I'm going to highlight this bottom picture down here. It's probably the best one. So where I'm drawing the yellow line, this is going to be our basement membrane, what we call the basal lamina. So it is separating, it's that connective tissue. You can go back to our like desmosomes and hemidesmosomes, helping to hold that bottommost epithelial cell to that basal lamina, the clear layer and the dense layer. That's the basal lamina, that attachment point of your epithelial cells. It delineates or separates the epithelial tissue above superficially to the connective tissue deep, right? So the basement membrane is that divider between connective tissue and epithelial tissue. So from here to the free edge, right? So I'm going to highlight that towards the top. They're just calling it the surface, but remember we now know that as the apical surface. So between those two yellow lines, we have many layers of flattened cells. And then this is just a scanning electron micrograph showing the surface of what some of those squamous cells look like. Kind of look like scales almost. So within these multiple layers of squamous cells, we are going to divide them into their own strata. So strata is just another word for layer. So depending on where you are looking at your skin, you will have either 4 or 5 of these strata. If you are looking at the palms of your hands, I'm not going to bring my foot up, but the soles of your feet, this is what we call thick skin, and there are 5 strata associated with thin skin. If you look anywhere else, like arms, legs, hands, face, neck, back, front, butt, those are all going to be thin skin, and they're only going to be 4 layers. So I will talk about the differences between thick and thin skin as we go through these different strata. So I'm just going to start from the bottom and go up. So the first layer is called the stratum germinativum. It is the cells that stick right on that basal lamina, so they're going to have the hemidesmosomes anchoring that stratum germinativum, or I think it's now called basal. This is an older picture from an older book. So it's the same thing. Stratum basal, I think that's how the lab manual calls it, and the text, the newer editions of the text might call it that as well. So the stratum basal is your deepest layer of epithelial cells in your epidermis. They are actively dividing, actively growing, and one of the main reasons is because they're very close to the dermis, which is right here. Well, I'll just highlight it because it's already there, the dermis. Dermis is connective tissue, loose connective tissue, which is very vascular. So these cells at the very lowest layer of connection have good blood supply. So they can get oxygen and nutrients, and they can get rid of their waste products, and so they're happy. They can grow and divide, which is great because that's their job. Germinate, germinativum, basal, base, right? So those words kind of give you clues as to the function and the location of these particular cells. So these are the cells that are constantly going through mitosis because, as we'll see when we get to the surface, we are losing those dead flattened cells all of the time. So, so we don't slough our cells all the way down to the dermis. We have to constantly be replacing, like a conveyor belt, these new cells that are coming from the stratum germinativum. All right, the next layer is called stratum spinosum. Right, so you kind of see this little bracket. It's about that much. So it's quite a bit thicker group of cells. Stratum spinosum cells are also living. They're close enough for diffusion to still work. So even though they're like one layer removed from the dermis, they're still enough. Cells are really small, so it's not a huge distance for diffusion to happen. So stratum spinosum cells are still living. And then we go into the next layer, which is a stratum granulosum. By this time, the cells are getting a little bit too far away for diffusion to take place. They are starting, the cells, as they're progressing up through this journey from the basal lamina, the basement membrane, to the surface, they're changing their characteristics. So they're really active in dividing in mitosis in the stratum germinativum and stratum spinosum. As they get closer to the more surface stratum spinosum layers, they're starting to really jam-pack their cells full of keratin and other proteins, waterproofing proteins, strength proteins. And then by the time we get to the granulosum, the cells are starting to die. They're dehydrating. They're not being able to exchange with the blood supply down to the dermis as well. And in this process, they fill up with all of these granules. When we go and stain them for the microscope, they kind of stain dark. So let me highlight, let me get the highlighter here. So we can see those stained cells right here. That's the stratum granulosum. And you can kind of see it right here in that. It's a little bit harder. You can kind of see it right here, that darker line. So that's the stratum granulosum. It kind of is highlighted, and we'll be able to see this in the microscope slides in lab. The stratum granulosum is a dark staining band. Oops, get rid of those highlights. Okay. So that's our stratum granulosum. The next layer, this is where we're going to differ depending if you're talking about the thick skin on your palm or the thin skin everywhere else. In thick skin, we have what's called the stratum lucidum. Okay, lucid means clear. So here's our stratum lucidum in this micrograph. You can see that clear kind of whitish layer that sits just more superficial to the stratum granulosum. So this is our stratum lucidum. It's just an additional layer. It's not super thick. It's not what makes thick skin thick. It's just an additional layer. As the cells move from the stratum granulosum into the lucidum, they lose all those granules, as we're preparing to move into the last layer, which is what we call the stratum corneum. Okay, so between these micrographs. The stratum corneum is the most superficial layer of cells. By the time the cells have made it to the stratum corneum, they're totally dead, they're totally dehydrated, but they're packed full of keratin. They're held tightly, very close to each other with those desmosomes, those button desmosomes from cell to cell. And so this is a nice thick layer of dead keratinized cells to give you that protection that you need. So you're going to find stratum corneum on all parts of your cutaneous membrane, but take a look in this picture up here. Look how thick the stratum corneum is here compared to here. This is the difference between thick skin and thin skin. Yes, thick skin has a stratum lucidum. It's an extra layer, but stratum lucidum isn't really significant in the thick part of thick skin. Look how thick that stratum corneum is. So that's what we have here. Okay, thick skin. Think about what you do with your hands and your feet. You're handling life with them all of the time. So that structure and function idea really plays out very well in not only all of your tissue types, but also when we start putting those tissue types together to make organs, like your integumentary system. You don't want simple squamous epithelium here. It wouldn't protect your body and your skin and your fingers from handling stuff. But nice, thick stratified squamous epithelium does a beautiful job. And we can slough cells off and not worry about sloughing all the way down to the vascular connective tissue underneath. All right, so those are our layers of the epidermis. You should know those. All 5 in the thick skin and the 4 in the thin skin, and the particular order from basal lamina to surface, from surface to basal lamina. Make sure you know those terms. In this picture here, just a couple of things before we move away from this slide is we're going to see it a little bit more with fingerprints coming up here, but we have the interface between the dermis and the epidermis is a lot of times, like I highlighted, the boundary, this basal lamina, it is very wavy, right? So it's got squiggles. Squiggle, squiggle, squiggle. And we can see the squiggle, 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 squiggle. We can see the squiggle, squiggle. Lots of squiggles, right? Those squiggles have names. They're called the dermal papilla. Papilla means bump. We're going to see that word a ton in this class. So dermal papilla are little bumps of dermis that are projecting up into the epidermis. And then on the flip side of that, the ridges that are coming down. So if I wanted to highlight down here in this picture, so the red is kind of on the boundary of the epidermal ridge, and the yellow is on the boundary of the dermal papilla. They're the same boundary. So dermal papilla, epidermal ridge, I guess you can think of it like that. Now you might be saying, well, why? Why are there bumps? Well, what we need to understand is the epidermis is connected to the dermis via that basal lamina, those hemidesmosomes. So if you had epidermis sitting on top of dermis right here, and this is the only amount of surface area that you had to make connections, that limits your amount of connections to hold your epidermis onto your dermis. But if we had our epidermis like this, stuck to our dermis underneath, where we followed all of those boundaries, we've just increased the surface area, allowing for way more connections. So that almost like an egg carton, the foams that those camping pads that have little egg bumps all over them. Imagine 2 of those like over each other. That's a really good increased surface area for connection. That's going to hold the epidermis very, very tightly onto your dermis. So structure and function shows itself yet again in our bodies. All right, so a closer look at those dermal papilla, and here they're calling it an epidermal peg, but it's the same thing as an epidermal ridge, right? So here is our basement membrane. I'm kind of highlighting in yellow, following that pattern. So that is the boundary between our dermis and our epidermis. And so you're going to have all of those hemidesmosomes connecting and all that kind of fun stuff. Now, just a quick little review. Can we identify the layers of epidermis that we just covered? So this is going to be, I'll do it in green. So here's going to be our stratum basal, right? And then we have this section right here. That's going to be our stratum spinosum. And then we have this dark staining band right here. That's going to be our stratum granulosum. And then we have this kind of clear layer right here. That's going to be our stratum lucidum. And then we have this big, thick layer here. That's going to be our stratum corneum. Excellent. Good review. So all 5 of those layers, this is clearly thick skin because we can see that thick stratum corneum. And if you'll notice where those dermal papilla and the epidermal ridges are, the thick part of that stratum corneum gets into these little folds. That's your fingertips. Fingerprints. Yeah, that's not going to focus, is it? But you've got your fingers, right? Look at your fingers. Look at your tips of your fingers, tips of your toes. You have the pattern, which is genetically predetermined based on your pattern of dermal papilla, as well as environmental. So this is the one thing, as far as I know of, that identical twins do not share. They have the same genetics, but they have unique fingerprints. So that lets you know development in the womb plays a part on your fingerprints. But once you have your fingerprints, they don't change throughout your lifetime. So you have your fingerprints, it's the pattern of how those epidermal ridges grow and the stratum corneum kind of folding over those ridges that give you your fingerprint patterns that you will have for the rest of your life, unless you lose fingers or toes or you damage your surface of your skin so much that the fingerprints are destroyed. As long if you heaven forbid, you get trauma and lose all of this tissue, you're not going to have fingerprints anymore, but what you will have is scar tissue, which will also leave a distinct pattern. Okay, so those are just a continuation of our discussion of epidermis. The next thing in our epidermal discussion is color, skin color. And I think, let's see, looking at my notes here, there we go, skin color. So there's 2 major pigments that are associated with the color of our skin. The major pigment that is biologically determined is melanin. So melanin right here. So melanin has a variety of colors depending on your genetics. So there's can be dark brownish black, it can be lighter brown, it can be reddish, it can be orangish. So that gives us the gradation or the gradient of skin tones that we do see. It is produced by a cell called the melanocyte, which looks like a big brown octopus, and it sits kind of smashed between your basal lamina and your stratum basal cells, right? Those basement cells. And it sticks these little tentacles up in between those stratum basal and stratum spinosum cells, and it is a manufacturer of melanin. So it makes the melanin pigment, it packages it up in these little packages called melanosomes, which then go and distribute themselves inside of your keratinocytes, right? The cells that are making the keratin, which is just your stratified squamous epithelial cells, right? Cyte means cell, keratin making cell, keratinocyte. So then if you look at where those little bits of melanin arrange themselves in this picture, you might notice that we have little packets of melanin hanging out over the nuclei of your keratinocytes. The job of melanin is to absorb ultraviolet radiation. It is your sunscreen for your skin's nuclei because it is a mutagen. Ultraviolet light mutates your DNA. And if it mutates the DNA in your skin cells, it could lead to skin cancer, which I have a mini lesson. We'll talk about that later. But that is the job of melanocytes, is to produce the melanin, to get distributed in those lower layers of cells, to cover your nuclei, to act like a little parasol or sunscreen, to absorb the ultraviolet radiation before it gets to the nuclei of your skin cells, to keep them from getting damage to the DNA, which could lead to skin cancer. So I have a couple of pictures up here before I talk about the carrots, is a variety of skin color. So why do, we have in our global population, we're all the same species, Homo sapiens, but we have individuals from very dark skin complexion to very light skin complexion. We also have people who, may not have any skin color at all, which we would call an albino. So this person is clearly of African descent, but no dark skin coloring. So what's going on here? Why do we have different colors of skin depending on, where you come from in the world? Well, the biological difference is not the number of melanocytes you have, but the amount of melanin that is produced and how how long the melanin stays in your tissues, and how far up it goes. So all humans have about the same number of melanocytes. Even individuals who have albinism, they have melanocytes. They just don't produce melanin. That's the recessive, homozygous recessive. So you remember some of your genetics from Y211 is when you are homozygous recessive for the ability to produce melanin, you don't produce any melanin. So wherever you would normally have melanin, in your skin, in your hair, in your eyes, you don't have any. And so you just are the color of what your tissues are. If you are on the lighter spectrum, you will have the same number of melanocytes as everybody else, but you don't produce as much melanin. And the melanin may only go, so far up into the layers of stratum spinosum, and it might get recycled very quickly. If you're on the dark end of the spectrum, you might produce a lot more melanin, and the melanin might go way higher and stay longer in your stratum. It might go all the way up to stratum granulosum, all the way through your stratum spinosum, where you wouldn't see that in a lighter skinned individual. So we have a huge variation and a variety in the human population of not the number of melanocytes, but the production quality, the synthesis rate of melanin, and how long it persists in your skin. Now, individuals who are on the lighter end of the spectrum, I am one of those. When I am exposed to the sunlight, what happens to your tissues is you produce more melanin. What do we call that? We get a suntan. You darken your skin tones, and that's going to be true for all individuals. All melanocytes are going to respond that way, but it's a little bit more prevalent in individuals who start off with a lighter skin tone because you can see that darkening more than somebody who is already on the darker end of the spectrum. And so it is a clear response to you exposing your body to the sun rays. Your body's like, whoa, too much ultraviolet light. I'm going to make more melanin. I'm going to cover up my nuclei to protect from ultraviolet damage. And then it turns winter, or you don't go out in the sun, and then your suntan fades. Well, it's supply and demand. We're not exposing our body to ultraviolet light. So the cell's like, I'm not going to waste all my energy making melanin when I don't have to protect the nuclei as much, because there clearly isn't as much ultraviolet light. All right, so that is our skin color due to melanin. This is a genetically predisposed condition based on your inheritance of your alleles for skin color. It's all kind of a genetic combination. The carrots, on the other hand, this is more an environmental impact on your skin color. Orange colored veggies like carrots and squash and things like that, they have a pigment called carotene, right? So carotene is an orange colored pigment that if you consume a lot of it, it actually stays in your tissues and you can take on an orangish hue. So if you like to juice carrots, or you eat a lot of carrots, or when my son was little, I'd blend up the carrots and he would eat a ton of carrots and butternut squash and stuff. And he kind of had orangeness to his skin, but it was a temporary thing because when we wouldn't feed him so much carrots and butternut squash and orange things, the orange tint went away. So that's a temporary thing. So melanin is more of a permanent genetic trait for your skin color. Carotene is a temporary thing based on your diet, basically. There are a few things that could affect your skin color. We have dermal circulation. So here in this picture, we can see that these fingers are turning bluish. This is what we call cyanoses due to a lack of blood flow. So there could be something going on. There is something going on with that patient's hand where those two fingers are not getting the proper blood supply. And so when you don't have the red blood circulating through your tissues, you look kind of purpley gray. That's kind of the color of our tissues without blood. This is supposed to be the picture of an angry person. Emotions is what I was trying to capture with that. Embarrassment sometimes is you flush, right? Blood circulation. You can vasodilate, or when you're hot or when you're cold, the changes in your dermal circulation pathologically, is that the right word? With a disease or a disorder, or just transiently with changes in emotional states. Now, this picture here is trying to show you the yellowing of that person's whites of their eye. There's a condition called jaundice. You may have heard that before. It's where your liver cannot process a biological waste product called bilirubin. And bilirubin is a yellowy color molecule. And if your liver is not working properly due to sclerosis or hepatitis conditions or liver damage or liver cancer, then your blood builds up this bilirubin pigment, which then kind of diffuses into your tissues, and then you take on a yellowish hue due to the buildup of bilirubin. This last picture is a genetic autoimmune disorder called vitiligo, where your immune system actually attacks and destroys your melanocytes. So it could be random. There could be some genetic factors to it. Autoimmune diseases are still kind of in the mystery category for a lot of medical research, but for whatever reason, the melanocytes are destroyed. Wherever the melanocytes are destroyed, you cannot produce melanin, so the outcome is kind of a patchy appearance. It is more obvious in individuals who have a normally darker skin tone because there's going to be more contrast. People who have a paler skin tone who are not producing melanin, you might not see quite as big of a contrast between the normal pigment and the parts where the melanocytes have been destroyed. Okay, so that's our skin color discussion. And then lastly, to wrap up our discussion, I know this is a little bit of a longer mini lesson. It's not super mini anymore, but there's a lot of stuff going on with your epidermis. This last slide is talking about how our epidermis, the keratinocytes, stratum spinosum, primarily keratinocytes in our skin, are responsible for making one of our vitamins, which, because we can make it, means it's technically not a vitamin. A vitamin is usually something that we have to consume in our diet. And we do consume a lot of vitamin D in our diets, but we do have the ability to produce a good amount of it. And if we don't, then we can be vitamin D deficient and have to supplement with external sources. So here's the pathway. And it seems a little bit counterintuitive because we were just talking about how UV light is dangerous to us because it can cause skin cancer and mutate our DNA. However, a little bit of UV light is good because what it does is it triggers this whole process for your body to manufacture vitamin D, which is why I will show you why I have a picture of this bow-legged child x-ray up here. So in our skin, there are precursor molecules in those living stratum spinosum cells. You don't need to know this term, but we'll just call it the precursor. It is a cholesterol-based molecule, so it's a lipid-based molecule. In the presence of ultraviolet light, it is converted into vitamin D. You do need to know this one. Maybe I'll highlight the words that you do need to know on this picture. Cholecalciferol. So cholecalciferol is the anatomical or medical term for vitamin D3. Okay, so this is what we get from our skin exposed to UV light. We can also take in vitamin D in supplements or meat and fish. So some animal products have high levels of vitamin D3. You can take that from eating them, or if you don't like fish or meat, you can get your vitamin D2 from supplements. All of those sources of the cholecalciferol, vitamin D3, goes to your liver. The liver processes it into another intermediate. Again, you don't need to know the name of this one. We'll just call it another intermediate. And that intermediate then goes to your kidney. And then the kidney produces a hormone called calcitriol. So these are there's two key words that you need to take away from this, kind of like new vocabulary. Cholecalciferol is vitamin D3, and calcitriol is the hormone that's produced by your kidneys. The other ones we can just call a precursor and an intermediate, but you do need to know the pathway as well. So stratum spinosum cells to cholecalciferol, to the liver, to an intermediate, to the kidney, and finally calcitriol. So what's the connection between calcitriol and bone health? Well, glad you asked. No, so calcitriol is a hormone that when secreted from the kidneys goes and targets your digestive tract, like your small intestine primarily, and helps to absorb calcium from your dietary intake. So a lot of times you might hear, if you combine calcium with vitamin D, those two work really good to increase your calcium uptake. That's not just a marketing scheme. It really is the reason why you want to take vitamin D and calcium at the same time, because it helps you manufacture this hormone calcitriol, which is critical for your uptake of the calcium that you're taking in via your diet. And if you lack this, if you lack vitamin D or you lack sunlight to make enough vitamin D, you get what's called rickets. So rickets is bones that have not calcified properly, and so a lot of times you can see, like in this X-ray, they're bended. Bones typically don't have that curve to them. Your femurs, that's your femur and your thigh, and it could be We don't really see it too much now in the United States because we have vitamin D fortified milk, and most people in the United States get enough sunlight or vitamin D supplements in the food that they're eating. But you have malnourished individuals or people who live in very northerly or southerly latitudes where 6 months out of the year there's not a lot of sunlight. And so if they don't supplement with vitamin D, they could end up getting rickets, which could permanently affect their bones for the rest of their life if they don't get them either straightened out and then calcified. Because if they're still flexible when they're young and it's taken care of, then they might be able to correct some of those curvatures because of the weight of the body is pushing on these uncalcified bones and they get bendy. All right, whew, that's the epidermis. That's a long one. The other lectures aren't going to be quite as long. So, all right, I will see you next time when we talk about the dermis. Bye.